Hello everyone. Welcome to the next in a series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we're going to examine a particular type of cell reproduction, that being mitosis. Mitosis, by definition, consists of the series of stages a cell goes through in order to divide the nucleus. The nucleus, of course, contains the DNA material. The purpose for mitosis, the reason it occurs, is twofold. Firstly, it ensures genetic continuity by creating a copy of the DNA. That is, it makes sure the DNA is carried on by maintaining the same number of chromosomes from one cell to the next. It also is involved in creating new cells for growth and repair. Mitosis occurs within the body, or also known as somatic cells of an organism, the skin, muscle, bone, etc. In fact, mitosis occurs in all cells except the gonads. The gonads, of course, being the ovaries and testes. When mitosis is finished, the end result is that we will have two nude cells that are called daughter cells. And each of these cells will have the same number of chromosomes as the parent cell. Genetic continuity will be maintained. Chromosomes, these are the strands of DNA that carry out the genetic code. Remember, the genetic code determines your traits, your hair color, height, etc. Chromosomes are found within the nucleus of a cell. And chromosomes themselves are normally single-stranded, but they will become double-stranded during interphase when they're replicated. So there are distinct stages of mitosis. In order, these four stages are prophase, metaphase, anaphase, and telophase. An easy way to remember this is the following acronym: P, prophase, please, M, metaphase, mother, C, A, anaphase, another, and T, telophase, table. Please, mother, another table. Each of these phases has a distinct role to play. We'll begin with our discussion of prophase. There are a number of things that happen during the prophase cycle. Initially, the chromatin, and chromatin is simply fine strands of DNA in the nucleus condensed to reveal double-stranded chromosomes. So following interphase, the single-stranded chromosomes have replicated to form double-stranded chromosomes, and during prophase they condense to actually become visible. Secondly, the centrioles will move to the opposite ends of the cell. Third, the nuclear membrane starts breaking down and eventually disappears. And fourth, the centrioles themselves grow filaments called spindles and are now called asters. The following illustration diagrams illustrates what prophase consists of. Note that during prophase, the spindles have formed their fibers, the centrioles have migrated to the poles to form what are now called an aster, the nuclear membrane is starting to disappear, and the double-stranded chromosomes have now become visible. The second phase, metaphase, allows for the following to happen. The chromosomes line up at the middle of the cell. The spindles attach the chromosomes at the centromeres. And by definition, the centromere is the middle portion of a chromosome that holds the chromatids together. To illustrate, with metaphase, the spindles, as you can clearly see, are attached to the centromere in the center of each chromosome. The spindles run to the centrioles or aster, and the chromosomes themselves are aligned in the middle of the cell. The third phase in mitosis is anaphase. During anaphase, the spindles pull the chromosomes apart into opposite ends of the cell, and once pulled apart, these chromosomes are now single-stranded and are present at each end. Here we see the process diagrammed. As you can clearly see, the individual chromosomes, the single-stranded chromosomes, are being pulled to opposite ends of the cell. In short, they are starting to migrate to where they will become new daughter cells. Following anaphase, we now have telophase. And during telophase, the chromosomes uncoil and become chromatin again. The nuclear membrane forms around each set of chromosomes. The spindle and aster disappears, and cytokinesis begins. What cytokinesis is the process of dividing the cytoplasm of the cell to create new cells. In fact, the cytoplasm will be divided equally among the two new daughter cells. And this occurs when the cell membrane in animal cells and the cell plate in the case of plant cells pinches together in the middle and eventually closes off from one another. Again, you see this process illustrated in the following picture. Here you can clearly see that the chromosomes have begun uncoiling and recondensing to form chromatin. The centriole begins to disappear. The spindles have disappeared. The nuclear membrane is reforming around each set of chromosomes. And the cytokinesis is occurring across the middle. In the case of animal cells, it is pinching off. In the case of plant cells, a cell plate would grow across. So to close up our examination, our discussion of mitosis, I present the following definitions. Chromatid is in a double-stranded chromosomes an exact copy of DNA. The nuclear membrane is the membrane that surrounds the nucleus of the cell. It keeps its contents together. The spindle is a long fiber created from the aster that attaches to the centromere of chromosomes. It pulls the chromosomes apart. The aster itself is the star-like structure at each end of a dividing cell that helps pull the chromosomes apart. And the cell plate. And the cell plate is a plate of tissue that grows between dividing plant cells during telophase. The cell plate separates the two new plant cells. Cytoplasm is defined as the fluid of the cell. It's important to giving structure to the cell. The chromosome is a long strand of DNA, or deoxyribonucleic acid. It's a protein which carries genes to express traits. Gene is a small segment of DNA of the chromosome that's responsible for the expression of traits. Centrioles are cylindrical shaped bodies near the nucleus of the cell that becomes the aster during cell division. And we have chromatin. Chromatin are fine strands of DNA within the nucleus of the cell that makes up the chromosomes. To conclude, I present the following questions from previous public examinations. This one reads, which structure is indicated by A in the diagram below? A is referencing in the very center that holds the separate single strands together. The correct answer then would be B, centromere. Chromatid and chromatin are genetic material. The centriole is where the aster is attached. The correct answer is indeed B, centromere. Also from a previous public examination, it asks, during which phase of the cell cycle does DNA replicate? We are being asked about the replication of DNA. DNA has already replicated before mitosis begins. The correct answer then is B, interphase. Thank you for your patience. I hope you found this learning clip helpful.